CELLECTIS is a gene editing company. From the start in 1999, CELLECTIS was founded with the idea of modifying gene, meaning even at that time the term gene editing didn't exist, but the main goal of CELLECTIS was really to be able to modify gene and cure diseases. So that means all the projects that we are doing at CELLECTIS takes their roots with the gene editing technology. So everything starts with a gene editing tool. So actually we are pretty agnostic in terms of technology. And in the past 21 years, we have been tested many of the technology that has been developed. This is including TALEN, CRISPR, meganuclease, megaTAL. And at the end, we have selected the TALEN as the most powerful technology. Why? Just because it's the most precise, efficient, and accurate technology. And we think that it is the most adapted for the therapeutic use.
When we have a sequence of interest on which we want to design talents, we scan the sequence with a machine learning model that has been trained over the year with all the data we have accumulated, and it will determine the sequences that have the highest probability of having a good activity. This machine learning model uses both sequence features as well as epigenomic features to determine the highest probability sites. This process takes place in a matter of a few dozens of seconds. In a second step, we will look at the specificity of the design talents. For that, we will use a second model that will scan the genomic sequence, look at the homologous sequences, and evaluate the probability that these sites are cut. Once we have determined the best candidates, we will send them to the New York lab so that they can synthesize them.
Hello, welcome to CELLECTIS New York. Today we will show you in this facility how we go from lead talent candidate to next generation UCAR-T prototypes. Please follow me. Once we receive the designs for the TALEN that we need to assemble from bioinformatics, we then use the robot to assemble the TALEN from the starting material. We have the raw materials that allow us to assemble essentially any TALEN we want against any target we want. The robot that we use is amazing as far as I'm concerned. We do many different things with it, and this robot gives us a lot of flexibility and a lot of power to do things efficiently, quickly, and in larger scales than we were able to do previously. We assemble the TALEN, we produce them. Here in New York we use next generation sequencing, which we have here in-house, that we use to test the TALEN from start to finish, from activity through specificity to, in the end, pick the best one. And then from that we generate a large quantity of RNA to then pass on to the next person for future tests in whatever experiments they're doing.
So the innovation team spends most of their time in the BL2 lab, that means Biosafety Level 2 lab, where is the place that we are allowed to work with viruses, and is the place where we can modify these blood cells from the patients to express the CAR, which is delivered via a virus. So after we have incorporated the virus into the blood of the healthy donors, and specifically into the T cells, we use an electroporator to incorporate the TALEN into the cells, and once we bring it in, the TALEN will express and will be able to modify the genome and generate the modifications that we want, the modifications that will make also each of our UCAR-T cell products unique. And then once we validate that we have all the edits and all the modifications that we need, then we need to expand the cells, because that's the beauty of the allogeneic CAR-T cells, to start with a small number of cells and to end up with a large number of cells, so to generate multiple doses. And then when we know that from a small number of cells, after we modify them, we are able to get a large number of cells, is when we have our prototype and we are ready to send it to the next level.
Welcome back to Paris. We will now show you how our process and analytical development teams work hand in hand to prepare scalable production processes. And as a sneak peek, we will also show you our GMP facilities where CELLECTIS is now producing critical raw materials and the gene editing tools we need for our manufacturing.
Along the life cycle of the product, the development work comes in between the innovation and the final clinical manufacturing. Our main mission, together with the analytical development group, is so to start from the proof of concept that has been made in New York, delivering us the talon and the core construct, to put in place a robust manufacturing process to allow the production of UCAR T cells. So really the main mission of our group is to upscale robust methods, efficient methods that are also suitable for clinical manufacturing, so easy to implement in a GMP environment. And we decided to slice it down into different unit operations that can be assembled together, put one after another, and then we assemble them, of course, into a global process, what we call today some kind of a platform. Every time we try to use the same global backbone and the same unit operations. However, we also need to fine-tune them depending on the product. And that fine-tuning is very dependent on what the analytical method are telling us. At every step, we look at the cells, we test their activities, and so we are working really closely with the analytical development group because they give us all the methods that allow us, again, to make sure that what we do fit what we want to do.
In parallel to the development of the process, we are developing all the assays that will be able to monitor and characterize the quality of the UCAR T batches. Among others, we are developing identity, safety, potency, and purity assays, and these assays will be developed and pre-qualified according to the guidelines from the regulatory agencies. All these assays are performed using flow cytometry, and one of the tasks of my team is to put in place the antibodies panels that will allow this characterization. Eventually, this assay will be transferred and implemented in the QC lab at Raleigh in a more controlled and qualified environment. This assay will be finally qualified and validated, and then will be used in routine for the release of the GMP batches for clinical use.
The GMP Paris production, which is, I will talk about this more, it's the production of our raw materials, our starting materials. This activity is a GMP activity because we use those materials in cells which will then enter clinical trials. So therefore, it's important that the starting materials are made according to good manufacturing practices. This means we make them in biobenches, in isolators, and of course in clean rooms. As you can imagine, we started manufacturing some of those steps, the cell banks, the plasmids. Now the next step will be the mRNAs, and the next step, which we are preparing in parallel, are the vectors. So now those products will be transferred by cold chain to Raleigh. So this is very important that the temperature are respected and that during the transportation the low temperature needed for the conservation of these raw materials is respected, and that we can document that this low temperature has been respected all the way from Paris to Raleigh.
Hello, welcome to CELLECTIS Raleigh. In here, you will discover the new wave of gene editing and how we apply it to manufacturing. Come on in.
This Raleigh facility is CELLECTIS's new state-of-the-art biomanufacturing plant. It's designed to take the processes that were created by our innovation scientists in New York and in Paris and optimized and scaled up by our development team in Paris, and duplicate those here at GMP manufacturing scale to make product that can go to patients. The materials are received from Paris and entered into our inventory system, inspected and stored in the GMP warehouse or controlled storage temperature chambers. They require low storage condition for the talons and vectors, so we have a number of different types of chambers at different temperatures that are appropriate for different types of materials. So once the materials have been inspected, inventoried, and put in the warehouse, they are taken out of the warehouse and staged into manufacturing as necessary during the 3-week production process. So different materials come from the warehouse into manufacturing as needed during this 3-week process. I and the team here are very motivated to make this biomanufacturing facility a reality and a big success for CELLECTIS. We're going to have control of our own destiny by having this manufacturing facility and being able to maintain supply to patients that need life-saving therapies.
So construction has been a big challenge, but one that we've really successfully met. We started with a shell of a building, have renovated it almost completely to incorporate all of the clean rooms and isolator technology that we need to manufacture CELLECTIS's products in a very unique and innovative way. So CELLECTIS has used industry standard equipment and adapted it for our process, as well as designing equipment specifically for our cell therapy process off the shelf. And that equipment is made for cell expansion, cell washing, cell selection, and then some of the custom designed equipment we use for doing the electroporation process to do our cell engineering. So the security of our cells is critical. Throughout the process, we make sure that we have chain of identity managed, as well as manufacturing inside the isolators to ensure that we have produced the most safe and pure product for our patients.
To build a true GMP manufacturing facility, you have to be exacting and specific about everything. There should be no activity that you do in a GMP space that doesn't have a process underlying it. And in order to support that, you have to build a facility that allows for all of these things to happen in an efficient fashion. So we receive an incoming collection of frozen cells. We thaw those cells. We use a lentiviral vector to transduce those cells. Then we electroporate those cells along with some talon to remove a specific section of DNA. From there, the rest of the process gets really simple. We expand the cells, we grow as many of those cells to make as much treatment as possible. We purify those cells to select for just the appropriately gene-modified T cells that we want to infuse into a patient. Then we move to the robot. Here we have an automated system that fills a series of vials, laser seals the filling holes, caps those vials. We inspect them, we freeze it down, and then we store it long term until the first patient needs their treatment.
At this site, we don't just manufacture, we do manufacturing and testing. During the process, but most importantly, at the end of manufacturing, in order to know that the batch is safe and efficacious, we have to do a battery of tests for release. The certificate of analysis lists all the tests that are required, what criteria they have to meet, and what results did you get. So after QC does the testing and assembles all of that information in the certificate of analysis, QA will review that and determine that the batch is acceptable or not, and they will release the batch for use. So it can't go out the door unless it has been tested and passed all of its quality control tests. If we don't have the QC tests that show that we meet the requirements of the certificate of analysis, we don't have a product. These products are the most challenging that I've ever worked on. The assays are the most complex. The products are the most complex because they're living cells, so it's not just a single molecule or some inert thing that can be highly variable and complicated to assess. I've always worked in science, and most of the time I've either worked in rare diseases or in serious illnesses where it truly impacts their life with the products that we make. And I found that never to be more so true than when working with gene therapy, cell therapies, and tissue therapies, because these products are life-saving products.
We've been really excited to have toured you this week through our CELLECTIS innovation cycle. We are innovators. We break paradigms. Our goal is to revolutionize molecular medicine in the 21st century. This is our commitment to a cure.
Nobody will explain you what the next wave of CELLECTIS gene editing technologies is. Come and see for yourself. Join us tomorrow.